All right. Well, good afternoon, everyone. My name is Calvin Chow. I'm the Senior Vice President of Medical Affairs at Tempus AI, and I'd like to welcome all of you to our webinar for our life sciences clients and other academicians, maybe also on this call. We are going to today discuss the topic of immunotherapies. And our title is Advancing Immunotherapy, Immuno-Oncology Therapeutics, the Convergence of Novel Biomarkers and Combination Strategies. And I'd like to call out that should you have any questions during this program, you may use the Q&A icon down at the bottom, and we will field those at the end of the program, time permitting. And as well, this program is being recorded. I'd like to start off by talking a little bit about Tempus's business structure and model. We are a diagnostics company for sure, but we are also an artificial intelligence database company that partners with life science biopharmaceutical companies in a variety of different ways. And this illustration shows you the variety of different services that we could potentially support. These are integrated solutions leveraging AI at many of these steps that can help a range of problem solving that you may have from novel target discovery and biomarker development in the earliest phases of both preclinical and clinical trial investigations, as well as through the execution part of clinical trials, conducting studies if needed, data management, supporting patient enrollments, companion diagnostics development, and even once launched or preparing for launch, supporting precision medicine adoption into a space that is increasingly relevant for companion biomarkers to be performed in addition to knowing which patients to treat for therapeutic agents. So today I'd like to introduce our speakers, and I'll ask each to introduce themselves. So Doug, would you like to please introduce yourself?
Sure. Hi, everyone out there. Doug Palmer. I head immuno-oncology at AstraZeneca. I also run a sort of a DARPA-like program called Advanced Translational Programs, which is geared towards building technologies and innovations to improve our oncology challenges, our target oncology challenges. Prior to coming to AstraZeneca, I spent about 2 decades at the National Cancer Institute working for Steve Rosenberg in areas around immune differentiation, activation, host interactions, and ultimately culminating in being one of the first CRISPR gene editing studies in immune cells in the US. I also here at AstraZeneca work a lot with real-world data, biomarker studies, in collaboration with Tempus to try and get to some of these key points that we're going to touch on a little bit. Glad to be here. Thank you.
Thank you for joining us, Doug. And Natalie Vokes, please introduce yourself.
Thanks, Calvin. My name is Natalie Vokes. I'm an assistant professor of thoracic oncology at MD Anderson Cancer Center. I focus in particular on computational oncology, so leveraging molecular data sets and using that to try to understand differential therapeutic response and resistance. And I have a particular interest in doing that in immunotherapy and trying to understand the biology of immunotherapy resistance, immunotherapy biomarkers, and I also coordinate our immunotherapy clinical trial space here at MD Anderson.
Excellent. Well, we'll tap your expertise here for sure. Tim Taxter, would you like to introduce yourself?
Absolutely. Hey, Tim Taxter, Executive Medical Director here at Tempus. I'm a pathologist by training. I've been at the company essentially since the founding, and so have been involved in basically all the aspects of building out our clinical testing services, supporting life science companies. And then really the last 4 years have been specifically focused on building out our AI applications products. And, you know, and for this talk specifically, we just launched our immune profile score test, which is an example of one of the assays that we include in our AI applications offerings.
Very exciting, and we look forward to some discussion about that. Thank you, and let's go through now the agenda. We'll first talk a little bit about, in general, the immuno-oncology landscape And we'll talk also about novel immuno-oncology biomarkers with particular attention to those that are on the market and those that are emerging, and supporting some of the existing biomarkers that could be considered for development purposes. We'll then finalize our discussion around combination strategies and some of the considerations that one may need to have as one embarks on potential development plans, looking at these combinations and patients' outcomes with them. And then finally, we'll have some Q&A at the end. All right, to start off with, I think that it's very clear this is a very rapidly growing market. This graphic shows you that from 2024 to looking forward, we can see that this market is doubling in size about every 3 years. And so with almost 4,000 active agents in development, and you can see how much is being spent on this segment of the market, it is definitely a rapidly growing area in oncology, and I think we should expect to see a lot more in the years to come. Next slide. And I think it's interesting that a very recent advisory meeting held by the FDA in September of this year came out with this conclusion from the advisors. This was an FDA ODAC voting that really weighed against the obvious benefits compared to the risks for patients with metastatic HER2 negative, MS stable gastric and gastroesophageal adenocarcinoma with a PD-L1 expression less than 1. I think this is interesting in the sense that there were drugs, both nivolumab and pembrolizumab had been approved by the FDA, I think in previous years, that did not necessarily take into regard the PD-L1 expression, but since then there had been accumulating evidence that there appeared to be some predictive elements to using PD-L1 as patients with lowest expressions under 1. It was not clear if there was a benefit, and there were certainly some adverse events. I think this speaks to the importance of biomarkers in general, but it also, I think, is a nice case to understand the limitations of PD-L1, as it seems to be the main biomarker that is being utilized today. TMB is also used. But as one looks forward in developing a new immunotherapeutic compounds, perhaps one needs to look at other potential biomarkers. And so, Natalie, let me ask you about some of the broader strategic considerations that one needs to think about today if one has an immunotherapeutic agent in development. What are some of the considerations for when a biomarker should be considered, and how might that be considered in the development of data for new compounds and immunotherapy?
Yeah, thanks, Calvin. That's a important question, and kind of I think this ODAC ruling is certainly an interesting case study, as you said, in a very heterogeneous biomarker approval landscape for immunotherapy. I think what we've seen in the development of immuno-oncology, in particular with these PD-1, PD-L1 inhibitors, is that PD-L1 has continued to be the biomarker everybody wants to beat. I think the analogy in lung cancer is second-line docetaxel. None of us think it's that good, and yet it continues to be the standard. And I think what we've been seeing with PD-L1 is that we know that this is an imperfect biomarker. I mean, the improvement in kind of predictive power over random chance is something like 10 to 20%. And what that means is when you deploy that clinically, it'll probably in some trials capture a responding patient population, and others it may not. Where that biomarker cutoff needs to be is probably different in different diseases. And I think some of that explains some of the heterogeneity that we're seeing in terms of the kind of outcome association, the trial results, and the approval landscape. And I think, the flip side, we're talking about a poorly performing biomarker, but the flip side is that our immuno-oncology drugs are also imperfect, and they don't benefit all patients. And without a biomarker, and without a biomarker plan in your trial design, there's a good chance you do risk the possibility that your drug ends up not being approved or not having a successful trial. And I think after the wake of the initial approvals that we've seen, in particular with lung cancer, we've seen a lot of negative trials, and there hasn't been a lot of investment in incorporating biomarkers into those trials, I think with the hopes of achieving a blanket FDA approval, but with the result being that we learned very little from those trials. So I do think that going forward, particularly when we have diseases where you have a regulatory landscape where we already have PD-1 inhibitors improved, it is important to incorporate biomarkers into our trial design and to be thinking really critically about what biomarkers we want to go after. I think we've kind of focused on single feature biomarkers like PD-L1 and TMB. The reality is that these drugs work at the tumor immune interface, and so we need to be capturing biology that's relevant to the tumor, the immune system, and how they interact. And there are a lot of ways to think about capturing that, both in terms of novel sequencing technologies like single cell, and also applying AI to existing data like pathomics, radiomics, and then building more sophisticated models that incorporate multiple of those features together. That is not going to be easy to deploy in clinical practice right away, but I think that structure needs to be built into the clinical trial so that we can do those analyses as we go and learn more as we go, rather than kind of consigning these drugs to chance with the hopes that it happens to work in enough patients to be positive.
Yeah, thank you. It certainly is a complex landscape, and I think we've certainly learned a lot over the past decade or so, and perhaps this next generation of trials for the biopharmaceutical industry will reflect some of these potential strategies one can take. Doug, let me turn to you and ask you, from a pharmaceutical company's perspective, I mean, when should one consider the integration of biomarker considerations in the preclinical and the clinical phases of drug development?
Thanks, Calvin. Yeah, I mean, I echo a lot of what Natalie was saying. I think, a lot has been spent in finding biomarkers. I don't think we've gotten bang for our buck out of them. I think in the non-IO space, there's certainly a lot of work going on for these targeted therapies, ADCs. We recently put out a thing around how you do AI digital pathology type approaches to measuring different types of targets like T-cell engagers or CAR T targets. So I think, and even those aren't even that straightforward, right? Even having the right target there doesn't necessarily guarantee you success. So I think, for the IO space, just when we start thinking about IO combinations and we start to think in how do we do these target approaches in combination with sort of an IO agent. I think a lot of it is, the broad sort of labels are gone. I don't think for IO that's going to happen anymore. Obviously, you can see it from the ODAC ruling. But also, when we think around, later into the market drugs, finding a niche, like where do they actually have the right benefit? So, I think we certainly have invested heavy in that space around biomarkers, and also working with different partners, CROs, and that have built out companion diagnostics. Because it's only finding a biomarker is one thing, actually deploying it is something completely different. And also moving towards things like AI path solutions, in terms of looking for, who has responses, who doesn't. We spend a lot of time building out human models. So, taking patient-derived platforms, pressure testing our drugs, doing some of the things, the sort of high HD oncology type approaches with spatial single cell, to trying to delineate what a good biomarker is. And then how do we go from sort of the research piece to the development and then ultimately to the implementation? Because that is, innovation is only as good as implementation. So, working with partners to how to get there is a lot of what we're doing. And then also, as a company, we want to help the high unmet need. If you think around how many patients do not benefit from, say, IO chemo combos that get it, how do we help those folks? So we're running, biomarker studies in standard of care patients, actually with Tempus, sorry, the disclosure alert there, where we're diving deep into looking at where patients are failing in these therapies, why they're failing on progression biopsies, which is, in itself is very difficult. and we work with partners, academic and clinical partners across the U.S. to to generate and gather this type of information, and actually working on, just a plug for me here, and work with those groups of people to disseminate that type of information. And this is very high-plex. I mean, we're very much into the computational space. How do we use foundation models, multimodal models, clinical transformer models to drive meaningful insights from things like spatial biology, from single cell, from ctDNA, radiomics, et cetera, to who's not benefiting and how can we help them, and also looking for surrogates of response. I mean, if we can answer earlier, what actually is a response, especially in things like neoadjuvant and resectable space, where the timeframes are much longer. So how do we get there quicker? So using things like PD markers and certain sort of biomarkers longitudinal to give us insights into what is and what isn't a response. And a lot of it is like is also understanding what is that sort of gold standard for us. What is our benchmark? What does success look like? And then how do we rate that? And I think that's also going to play into what is a good biomarker, and how we're going to leverage that. So I think it is sort of end to end. You have to think around from not just the research, but how you're going to deploy that. And we certainly think about it for a number of these solutions that I mentioned.
Yeah, so it sounds like, one needs to consider the biomarker development strategy simultaneously as you're developing your drug. They really now go hand in hand. Is that fair to say? Definitely,
yeah. I mean, with so many of our therapies, we have, selections. I mean, I was involved early on here at AZ for a pancreatic study, but biomarker selection for immune type assets, and how do we select the patients that would likely best benefit.
Terrific. Well, that's a great foundational discussion for the rest of our program here. This is just a quick snapshot of the number of drugs in a variety of different drug categories by mechanism of action. This is actually all drugs, both clinical and preclinical development. And you can see comparing 2023 to 2024, this is rapidly increasing across a number of these categories. The top category of over 3,900 this year, the immuno-oncology therapy, very nondescript category, reflects the fact that there's a lot still yet to be classified in the more specific categories listed below. And many of these are still in a preclinical phase. So next slide, I think really speaks to the complexity of the immune system's ability to eliminate cancer. And it really does require and rely on a number of different steps, starting with releasing the cancer cell antigens, and having those antigens presented to dendritic cells, and having the T cells primed and activated, and then having those T cells trafficked through the bloodstream to those tumor sites, having those T cells being able to integrate into the tumors and allow the specific engagements to happen where the killing of the cancer cells occurs. Very, very complex set of processes here, but I think we can think about this cancer immunity cycle to look at where drugs apply themselves, as well as potentially we'll talk about where mechanisms of resistance may arise. But we'll kind of look overall at these three broad sets of challenges. Number one, ineffective predictors. I mean, we have PD-L1, TMB, and MSI high today. But what else do we need? What additional information can be helping us both in predicting both sensitivity and resistance, and really understanding, number two, some of these resistance mechanisms that could be occurring at various points along this cancer immunity cycle, and how can we overcome some of these immunosuppressive factors. And then finally, we'll talk about combinations, because it's very possible that much more efficacy can be gained by combining immunotherapy with other therapies, chemotherapy, targeted therapies, or different types of immunotherapy, or new classes of drugs such as ADCs. And so we'll talk about that. Next slide. So this just gives you a graphic of some of the currently approved biomarkers that we've talked about. PD-L1, microsatellite instability, tumor mutational burden, but really a lot of additional potential biomarkers are obtainable should one integrate those diagnostic platforms into the appropriate studies. So we have biomarkers that are available through tissue. Listed here are things like TILs or POL-E, POL-D, single cell RNA sequencing, HLA status, et cetera. I think there's a lot there. I think that some of the mention that you've made, Doug, about the spatial features that may have a play there as well. Circulating biomarkers, I think, in addition to the actual peripheral T cells and soluble forms of PD-1, PD-L1, CTLA-4, there's also just simply ctDNA platforms that today can assess tumor response monitoring, something that actually Tempus has through our standard liquid biopsy platform, XF+, and circulating tumor cells, as well as MRD platforms that increasingly we're seeing immunotherapy needs in the adjuvant or early stage cancer space that may play a role. And then finally, gut microbiome, which has received a lot of research attention, but also a lot of potential there too. Let me just ask you, Natalie, as you look at this sort of, all the realm of possibilities here, is there anything that really you feel stands out or warrants for particular attention to specific kinds of patients?
Yeah, I think we're, I think this figure is a little misleading because we're really only seeing half of the pie chart here for three small tests compared to the whole wealth of research potential. And I think that's, what Doug and I have both been emphasizing is how much more there is to learn in terms of thinking about immunotherapy biomarkers. I think, as I mentioned in my first set of comments, for me, I think what we need to have going forward is some readout of tumor biology and immune biology, and we need to have a way to capture both of those phenotypes. And I do think that needs to be assessed prior to the initiation of treatment, but then there may also be additional value from assessing this longitudinally. And so that's where I think, the world of circulating biomarkers and liquid-based assays will be very informative, since that's going to be much more feasible on a longitudinal basis. We do have immune cells in the blood, whether we are detecting the right immune cells or compared to what's in the tumor. I think there's still more to learn, but I think that's going to be a very easy to access kind of biospecimen source as we start to try to build these data sets. I think there's a lot more to be learned from the tissue. I think we're getting, the technology is really changing. We can do single cell spatial, we can do sub-single cell spatial, we'll be able to do protein. And I also think AI applied to digital H&Es will be very valuable. So I think all of that's coming, and it'll allow us to learn more from a single section or from a couple of sequential sections compared to needing to cut up a 20 unstained slides or more. But I don't see that as being very realistic for the clinic, and it'll be hard to build those data sets simply due to the cost. So I'm excited about the combination of kind of more in-depth profiling on your pretreatment biopsy using a novel platform, coupled with really investing in novel technologies applied to blood and thinking about it dynamically. So, assessing early on treatment response assessment, can we distinguish between different kinds of responders and resistance early, and then funnel those patients into different trial strategies.
Great. Doug, let me ask you some of the platforms that you feel are particularly promising in some of these newer biomarker arenas.
Yeah, it is quite a journey to go from from insights to action. And so, I think around things like, let's just take one and no particular importance, like AI pathology, right? It's been around for a while. The idea that you can scan an H&E image and you can think around, what is immune infiltrated or activated. There's a bunch of papers back from, I think it was like 2018 around this work. And then it's like, well, how do you actually use that? How is that actually deployed, not just in clinical studies, but in practice? And so I think there's been a real disconnect between even just something like that, which, everybody's H&Es are, from like the 1880s. So, it's been around for a long time. How do you deploy that in a way that is even in a clinical study, it's a real challenge. And so, I think building out these sort of robust data sets is something that, not everyone invests in. It's expensive. It might not be necessarily drug project related. When you think about the sort of clinical trial design, I mean, you're asking if does this therapy help these patients, not necessarily, why do patients generally fail standard of care? It's a much tougher ask. And so I think, I think that's where, we a lot of rush to get into market, but not necessarily having built in the biomarkers or even the assessments to, ultimately deconvolute that. I think one of the things that we've been trying hard to do is, how do we actually generate a place where we can where we can dive into these types of assessments, see if there's value in a robust way, and then think about how do we build that towards deployment. I think around like things like foundation models, a lot of excitement around them. I think that's really, the data is quite amazing. I mean, the fact that you can take RNA-seq and make like a a Dolly-like image for those who who dallied with that, and it can fool pathologists and pathologist friends of mine. They have no idea. We should just yesterday we're talking about it and showing a bunch of images, and they failed about 50% of the time detecting AI-driven pathology slides from an RNA assessment. But that's cool. But how do you actually make that useful, and how do you deploy that? And I think, that's where we're trying to work now, very much with sort of the later stage of drug development right off the bat, and say, okay, how do we actually go from discovery to development and deployment sort of in one swoop, and not get 10 years down the road to find out that one, this was not deployable, or it wasn't actually that useful. So I think for us, from our perspective, I think we've invested very heavy, not just in these types of assessments, but in clinically relevant types of samples, either from patient-derived platforms or biomarker studies, a lot of real-world data. And I know we'll talk about resistance later, the pros and cons of that. But I think that is an absolute challenge. But I think the way we're trying to kind of steer is to get it in late to understand what does success look like and work backwards.
Doug, you raise a very good point about deployability. I think that it requires that the platform be easily available, usable by clinicians in a very practical way. And so maybe that's a good segue to our next slide here, where I'm going to ask, if we flip slides. Okay, there we go. I'm going to ask Tim to actually talk about something that we've developed here using our NGS tissue-based platform, XT and XF. Tim?
Yeah. So yeah, we'll talk about our immune profile score test, which, I'll kind of reference the 2 points that Natalie and Doug just made, which were great in the sense of, like, Natalie was really emphasizing, like, going beyond, obviously, like the biomarkers we already know about. And in our case, the motivation was really to leverage our RNA sequencing assay to really maximize, the value we're getting out of that to understand immune functionality. And I also like Doug's point about, it's like, it's one thing to have the insights. It's another thing to actually translate those into practice. And, Tempus is pretty unique. We've actually had an immunology department, since I've been at the company for like 9 years, which is pretty unique. And so, we've been working kind of in the background on this stuff forever, as a lot of other groups are. But really, in the last couple of years, finally got to a point where we felt like we had a large enough corpus of data to really do something meaningful in terms of developing a pan-cancer immunoprofiling assay. And so that was really the motivation behind the work. And so we started with essentially a 3,000 patient cohort for development and for validation. And in terms of the features, just at a super high level, we started with, a lot of single gene or RNA signature features that are out in the literature, and including also some developed at Tempus, either using single cell sequencing technologies or some other approaches. And those were part of our Tempus IO platforms. That's a kind of a research and explore platform we make available. And then we, applied our machine learning methods to those features and ultimately ended up with the final model that includes TMB along with several single gene RNA features, some of which will be familiar, like CXCL9 or PDL1, in addition to RNA signatures such as MDSC, which some people might be familiar with, in addition to a couple novel Tempus signatures, one of which was single cell derived. And so that's what we took that IPS score signature, and that's what we then ended up taking into a clinical validation, which is the next slide. And we can happy to answer plenty of questions on these at the end of the talk if anybody wants to get into the details. But for our clinical validation, as I mentioned, we're thinking about this in the pan-cancer setting. And these patients were metastatic or advanced stage in the case of head and neck cancer. And they were all ICI treated either in the first or second line. And these were all cancer types with an ICI approval. And in that validation, we saw strong stratification in the pan-cancer overall cohort. And then we also did several tests to determine that the IPS signature was outperforming the known clinical biomarkers, and also doing lots of different subgroup analyses to demonstrate significance within specific cancer types, and across other clinical features. And now really it's kind of the next part of the journey. This is really sort of where it begins, and now it's working with collaborators for future clinical development efforts, in addition to how do we think about this in the evolving immunotherapy space, as the other panelists mentioned, in terms of like how do you think about this and in terms of like PDL1 plus other treatment strategies in combination, and what do those biomarker strategies look like. So it's an exciting time, and we're looking forward to working on this more.
Great, Tim. Let me ask Natalie and Doug, any questions from you as you look at this data, as well as kind of think about potential application of a signature like this, which could be run off of a standard NGS tissue test that we perform. If not, that's fine.
I'm happy to jump in. No, I mean, I think this is the path that we've been certainly excited about too. Is from how do you predict who's going to benefit, and also think about that patient journey, right? I mean, if you're not going to benefit from a drug, why waste your precious time, the side effects associated with that? I think that saves them a huge degree of effort and time wasted. So I love that idea. I guess the question is going to be like, how do you actually deploy that? I mean, we are not a diagnostic company, so we, working with partners for us is great, to kind of action these types of things. I guess the question is going to be, how, are these things going to be picked up? What's the market look like? And ultimately, for regulators, what is behind what is behind this engine, and how does it contribute? I mean, one thing I think around is, you ChatGPT, you can answer, anyone can be an armchair doctor, right, with ChatGPT. The question is going to be, how are you going to make treatment decisions based off ChatGPT? I mean, or are you going to make, treatment decisions based off of a sort of a black box algorithm? And I wonder how regulators are going to actually kind of think about this space. And I wonder if, if it's those conversations have already gone on, and what is that message back from the regulatory agencies?
Yeah, that's a good point. Natalie, let me ask you in general. So we have two curves here, obviously IPS high and low. One fares better in terms of overall survival from first-line checkpoint inhibitors than the other. For patients who are, let's say, in the low category, IPS low, how would you think about those patients, let's say in practice? Are these patients who maybe have a colder tumor environment, microenvironment, or do they need something extra in addition? Obviously, they were all patients who had some checkpoint inhibitor treatment, so we assume that they're within the indication. But how would you think about those patients?
Yeah, I think that's a great question. And I think that's one of the challenges to think about, to kind of focus in on as we build these kinds of models, to think about how it would actually change practice. In lung cancer, anybody who does not have a targetable driver would be treated with an immune checkpoint inhibitor. So knowing that they aren't going to be a great immune checkpoint inhibitor responder doesn't necessarily change the fact that I'm still going to give them a shot at it. And so I think linking these kinds of scores to more specific treatment decisions that I'm able to make would be very helpful. In lung cancer, and we touched on this a little bit, with the question about treatment selection. I think in lung cancer, we are choosing between pembrolizumab monotherapy versus chemo combinations versus CTLA-4 combinations. So if I knew, for example, that this signature identified patients who wouldn't respond to PD-1, but who in fact might respond to CTLA-4, which is what we've seen with PD-L1, then that would be a very strong rationale for me to think about augmenting their immunotherapy regimen with CTLA-4. I think the other patient populations to think about where this signature might change what we do clinically would be those patients where we don't necessarily give immune checkpoint inhibitors. So second line EGFR, second line ALK, those are patients who in community, we tend to treat those patients with immunotherapy in clinical trials, but I certainly know a lot of community oncologists will reach for the Empower 150 regimen. So knowing, hey, can we identify your EGFR IO responders, of which there are some, even if they're not that many, that would be very valuable clinically, or to kind of start to differentiate between these subgroups. I think the other kind of understudied patient population in lung cancer, where we haven't had the data to support the biomarker studies in academic centers, has been squamous. They don't get profiled as much as standard of care, but really understanding how these biomarkers perform within these subgroups within lung cancer would also be very helpful, and I think add nuance to the decisions that we're making clinically.
Oh, those are wonderful insights. Thank you very much. Okay, let's move on. So this is just another representation of the similar cancer immunity cycle that we saw earlier, but with attention to the resistance mechanisms and the different areas where resistance mechanisms can occur, from antigen recognition and presentation and the activation of the T cells to the trafficking of the T cells, their migration to the tumor sites, the recognition by the cancer cells by the T cells, and then finally the actual point where checkpoint inhibitors are acting to facilitate killing of the cancer cells. So here, I think that we really have a variety of thoughts, considerations for resistance mechanisms. Natalie, would you like to comment just about, where do you see, for at least for today's therapies, where are some of these, what are some of the considerations here in thinking about resistance?
I think that we focused a lot as a field on predictors of immunotherapy response, and I think we've had a much less focus on mechanisms of resistance. I think some of this is data-driven. We don't have the post-treatment biopsies. And so I think we actually have a hard time mapping what we observe clinically to what is happening biologically. And I think that's actually a major kind of unmet area of research need that I think many of us on the call today are working on. I think the way we divide this up clinically is often between intrinsic versus acquired resistance and kind of the temporality of when the resistance is occurring and whether it's a isolated site of progression or large scale progression. And I think that those nuances can inform a lot of our treatment decisions. I suspect what we're dealing with when we are making those categories is, some differentiation between our cold tumors versus our non-cold tumors. And then within that non-cold tumor and the acquired resistance setting, there's a lot more to understand about antigen loss and changes in T cell phenotypes and other myeloid populations and immunosuppressive populations that I think are coming in the later phases.
Great, thank you. Let's go to the next slide, which actually speaks to primary resistance. And so this is just a set of data that looks at a variety of studies. I think that curves here reflect some patients in the first line immunotherapy plus chemotherapy, or single or dual agent immunotherapies in the first line, and then those receiving immunotherapies in the second line therapies, typically following a first line chemotherapy. So it looks like that this primary resistance appears to be higher in these latter patients who are getting them second line. Be that as it may, I think that, it speaks to, I think, what you were saying earlier, Natalie, the need for much more research in understanding mechanisms of both primary and secondary resistance. But, are there really kind of general points that you'd like to point out here in terms of thinking about primary and secondary resistance in patients?
I think that, it's important to know that when we're talking about immunotherapy combinations and some of these options that we're seeing here, we are really talking about two different things. I think when we talk about immunotherapy and what we want for our immunotherapy patients is long-term durable disease control. And that's what immuno-oncology offers that's novel, and that's the goal. When we look at some of these response rates, that doesn't necessarily reflect that actually happening. And I think when we've looked at the chemotherapy IO combination data, a lot of what chemotherapy is doing is buffing up your initial rate of response, but not necessarily affecting whether you have an effective anti-tumor immune response over the long term. And I think the emphasis on kind of primary resistance and predictors of radiographic response is really important. We need to know who's going to respond or not, because if somebody has a high disease burden, you want to throw everything at them. But that doesn't mean that we're actually inducing an anti-tumor immune response. And I think we need to be really thoughtful about disentangling simply non-overlapping disease treatments, which is fine, versus truly augmenting our immunotherapy. And that tends to show itself more over time in the acquired resistance settings. And that's where I think some of these other combinations, even Checkmate 227, some of those curves look better over time compared to some of the chemo combination curves because it's an immunotherapy drug. So I think, just reiterating my earlier point, that the initial response rates are important. That's an important clinical decision, but I think we also really need to be focusing on the durability.
Excellent. Okay, let's shift over to discuss combination treatment strategies here. This is a very busy graphic, but you can just look at the on the left side, some of the existing approved immune checkpoint inhibitor combination partners that we have today. I think we've seen some mixed results here. I think you referred to this, Natalie, that certainly we have CTLA-4 and at least for the anti-LAG-3 agent in melanoma, there's been some success there, but there's also been some lack of success for some of these agents. I think the anti-TIGIT inhibitors were disappointing in combination, and I think over to the right you can see really a lot of the broad potentials that we have for other combinations here, ranging from ionizing radiation to CAR-Ts in solid tumors, potentially targeted therapies, looking at intracellular pathways, ADCs, a variety of immunosuppressive or immunostimulatory cytokines, gut microbiome, and cancer vaccines, and finally bispecific antibodies. But let me just ask, what are you most excited about here, Natalie? Or where do you think, there's a full range of possibilities here, but how might you look at this range of options here in terms of where might be the most promise?
I think certainly a lot of clinical trial activity right now around LAG-3. So we'll see about that. Certainly promise in melanoma could be coming for other cancer types. I think we're seeing a lot of the checkpoint-based strategies moving into the front line with the, on the backs of many negative second-line trials. I suspect they will have more activity in the front line. I think the challenge will be to distinguish to kind of identify the population of patients who wouldn't respond to the PD-1 who benefit from that additional drug. But I suspect there will be some margin of patients, like 5 to 10%, who do benefit from that strategy. So hopefully some biomarker work to be coming there. And then I think where we're starting to see some signal are some of the bispecifics, and I think we'll we'll talk about that. So kind of different combination antibody approaches in one molecule. And I think some early positive data from some of those drugs. And then, I think other kind of more cytokine-based strategies and cellular-based therapies may help alter the tumor immune microenvironment more effectively and augment the immune responses in ways that we're not getting just from the checkpoint inhibitors alone. So I'm excited about some of those trials.
Great. Tim or Doug, do you have any other comments about thoughts about combination strategies in general?
I mean, a lot of thoughts. How much time we have about combinations. But probably some I'll share here and some I won't. I think going back to resistance, right? Because I mean, obviously we can try any type of combination willy-nilly, but we know around, even just combinations, but the timing of combinations might be important, from, checkpoint blockade plus small molecule inhibitor studies that we run. We know that timing can be actually quite critical, and sequential isn't actually beneficial. So that's one extra little piece. I think also, understanding this resistance mechanisms. I think, diving into why are they, why doesn't this give you durable responses? We've certainly dived in deep in real world data. And what was interesting that we learned, some published, some not, was that it wasn't actually new pathways that we did. I mean, a lot of them were already we already knew. I think what was really exciting about it was the sort of the epidemiology of resistance, what patients were failing and why, and what that proportion of the population that was. For example, things like antigen processing, right? We would think that would be a big one. We actually did not. That was not a spoiler. It was not a big one compared to some of the other resistance pathways, which was really interesting. So, I mean, I think, it's a little bit more informed approach. And then the other thing is how do you actually test all these combinations? Because, I mean, it's expensive to do, phase 2 study. It's competitive, with all these frontline IO agents, who's going to be left? What's the patient, landscape looking at? That adds time. And also readout takes longer. So, how do we pressure test some of these combinations? And, a lot of what we've been doing and others have been how do we build better models? How do we go from mouse models from the 1950s, growing on the back of some nuclear reactor to something actually a little more clinically relevant? And how do we operationalize that? And how do we build confidence that those models are useful and reflective of the human experience? So, I look at all these, and I mean, I'm with you, Natalie. The bispecifics I'm also excited about. I mean, I might be biased, but but I spent 20 years doing cell therapy too, and I can see the durable responses that cell therapy can do. But the challenge in production, the challenge of treating, some of the toxicities associated with that, certainly ADCs have a huge resurgence right now. And now we're thinking, how do we model ADCs with other targets, from things like PARP inhibitors to IO agents, even cell therapy. You know what I mean? And so I think instead of doing 300 or 400 phase 2 studies, how do we actually de-risk some of these investments that we're doing, and spending a lot of time trying to get around that.
That's a great point.
One other thing too,
Go ahead, Tim.
I've always been a fan and following a lot of the radiation work and where it merges into the IO space, and I think that's one where I feel like I've seen in the last few years maybe a lot more thought around the timing of radiation and how to do that effectively, and also what are the biomarkers, especially on the resistance side and immune suppression side, that are informative in that space. And those can be much different than they are for PD-L1, for instance. It's a lot of work in looking at aneuploidy and those type of cancer specific markers. And Natalie, I know that there's a lot of work at your institute specifically focused on some of these novel radiation therapy trials. So I know that's always a space that I've been interested in, because there's really sort of a dearth of any kind of biomarker work in radiation oncology. And so I'm interested to see how that develops, especially as these new therapies come in line.
Yeah, so I think really we are we've been well served with really kind of maximizing how much information we can get through modeling to really de-risk the programs. I think, Doug, as you were saying, I think that's something that clearly Tempus has had people utilize its tools to help answer some of those earlier questions. This is actually just some data that was presented very recently at the World Lung Conference this year. Natalie, I don't know if you were there, but maybe you can comment on this. This was actually a set of data from Chinese investigators, I believe. Ivonescimab is a bispecific antibody that addresses both PD-1 and VEGF. And really produced, interesting data here against pembrolizumab. I think there was some criticism about whether that was the right control arm, but nevertheless, I think it was a interesting, large degree of benefit. But I think speaks to the potential for addressing, a potential mechanism of resistance, which is really getting the T cells trafficked to the sites, perhaps through inhibiting VEGF in addition to the PD-1 inhibition. Any comments on this, Natalie? Your thoughts about this particular strategy, as well as this where this might lead for non-small cell lung cancer patients?
Yeah, I think we were all very excited to see this data. I think this is one of the few positive kind of novel immunotherapy strategies we've seen in lung cancer in the last couple of years. And it was very positive, as you said, with the caveat being that this was primarily a Chinese population rather than a global population. And the company Summit is working to replicate these results globally. I think VEGF has long been a target in lung cancer, and we've seen over the years many positive trials with VEGF inhibitors, in particular for PFS. And we have seen many VEGF-based trials that have not shown an OS benefit. So I think it'll be very exciting to see the OS data, hopefully next year or soon, and kind of make sure that we're not seeing a VEGF effect. I think if we see OS, and we also see improvement in PFS and OS in some of the other patient populations that they're going after, including EGFR, I think that'll be very exciting proof of principle that we actually have kind of a novel strategy that seems to have legs. And I think that'll be a foundation for a lot more, combination strategies, certainly, but also biologically informed investigations.
Excellent. Let's, I think, make sure that we have time for additional questions here. Yeah, so let's just use the remaining time. I think one question I have for actually any of you, Doug, Natalie, Tim, your thoughts on drugging Tregs, regulatory T cells. Any thoughts there?
Interesting. It's funny because I remember back at the NCI 22 years ago, I think it was, we published a paper on Tregs in mice. I think it ended up in JI, and it was probably one of the most cited JI papers. I think it was almost 1,000 citations, something insane. And so it's amazing how long we've been talking about this, and yet I've yet to see something really come to fruition.
Okay.
Despite the thousands of Nature papers. So, I wonder, I know, and we know that it's complex in humans, things like FOXP3 just not the same as a mouse. So, I wonder, I think there is a lot of talk about it, people who have been working on it a lot. We've mentioned a lot of things here. Actually, we'll let Natalie talk about Tregs if she likes to talk about Tregs. But I mean, if you want to pass, that's fine. I think, for me, it's how do we prioritize? We've talked about a whole bunch of stuff, right? So when I leave this, what is my top 5 things I want to work on that I care about? And I generally like to, distill that from other folks too. What is the most important thing for you? I mean, we talked about biomarkers. That's a huge part of what we're doing. Early detection of response, that's really important for us. How to deconvolute the human experience and the human biology, that's a big part, using different tools. And then, how do we model? So those are, for me, the big ones. I'm not sure that one in particular has. I agree with Natalie around some of the things about VEGF. It's been around forever. I think these are exciting ideas. I wonder if we're missing something else. But in terms of prioritization, that's kind of where I'm at. But I don't know, Natalie, what you? take a moment to reflect.
No, I agree, and I think we actually, Doug, share a number of kind of overarching priorities. I think for me, Tregs, when we've looked in our data, I suspect that they are important, but not in all patients. And certainly as a resistance mechanism, it's not the population that we see, the cell population changing in all of our patients. But it may be important for some. And so I would love to think about patient identification strategies and kind of saying, okay, this is our subset of patients in whom Tregs are driving resistance, and focus the drug development on those, because I don't think it's going to be a bonanza, like an all-comers approach.
Mm-hmm. Yeah, and just call out to any of our audience, if you have any questions, please feel free to enter them through the Q&A chat box. I don't think we have any. Well, here's one actually. With so many IO assets in development, what needs to be done to help patients make informed decisions about their care? It's a very general question, but any comments there, Natalie?
I think I think we are entering a time right now where there are a lot of IO drugs being developed. It feels like they're all moving to the front line without necessarily a strong evidence base that any of them do work. And so I think participating in these trials will be important. Just as a blanket statement, I do think that that's going to help us advance the field. I think focusing on trials that have some biologic rationale, that have, if not biomarker stratification, then at least assessment built in, even though that's more of an ask in terms of tissue and blood, will be important, because it'll ensure that we're truly learning even from the inevitably negative trials that will read out. But I do think we are entering a time where there will be a lot of trials, and there will be a trial for everybody. It's just a matter of kind of participating in them. And I think in particular for patients who we think may benefit from augmenting an anti-tumor immune response based on the biomarkers that we have so far, those would be the patients who I feel I'm really excited about these kind of combination strategies, just try to do a little more for them.
And I think I remember hearing some. Oh, sorry. Go ahead.
Go ahead, Doug.
I guess have a conversation with your doctor.
Yeah, some statistic that I think it's about 5% of the people that are actually of the 100% of the people that are eligible are the ones that actually go on clinical studies. It's about 95% or something don't go on clinical studies that are eligible to go on them. So it's a pretty huge swath of the population that opts not to do that. Curious what those decisions are. There's also risk and safety and time. So, I mean, we've had plenty of friends who've gone through that, and it's a tough place to be as a patient.
Yeah, and a complex problem. Tim, any parting thoughts from you about the IPS score? It's brand new. It's just been launched. Any final comments you want to make there about potential applications of that to our life science partners?
I think Natalie was alluding to it. It's really, the next steps for that are follow-up studies to dive deeper into those specific clinical questions. Like Calvin, like you've been around for a long time too, right? Like the origin of how we initially were working on sort of like the version 0.5 was specifically focused on PD-L1 high lung cancer patients, right? We wanted to answer that question of who should get chemotherapy, or who to get pembro mono. And so, I think that's still like probably the most important, follow-on study we'd like to do in some capacity. And that may involve, trying to work with, alliance groups. And so I think for IPS, at least on the clinical testing side, it's really just about, moving forward with future studies. And then, on the pharma side, I think it's, likely going to be how can we use that as a possible foundation to leverage other biomarkers in conjunction with that. I've done some work also with, how do we think about cancer subtyping algorithms, things that are picking up like, fibrotic signatures like TGF-beta activation, and combine that with IPS. So I think to me that at least for the pharma world, it's really about like IPS plus, what other, biomarkers are relevant.
Well, thank you very much. I believe we're up on time. I do want to thank the three of you, Natalie, Doug, Tim, to learn more. You may use the QR code that you see here, or go to the website indicated here to obtain more information on the on this topic. And I do want to thank all of you. I think this has been a very insightful conversation. And obviously, there's a lot more work to do. So we hope to work with many of you that are listening here, and as well as if you have ideas on how some of our platforms may be useful in answering some of these questions or helping to model or de-risk some of the elements of decision making through your development process. This is something that we love to have engaged conversations in. So with that, I'd like to conclude and thank you very much for your attendance here. Have a nice day. Thank you.